Welcome to section 9.2, where we're going to focus a little bit more in depth on that M phase we talked about of the cell cycle. So the part where we're going to divide the nucleus up and divide the cytoplasm. So starting off, we've said mitosis is going to be nuclear division. So it's going to take us from having one nucleus in a cell to two. Now typically this will be followed by cytokinesis, which will then break it up into two cells, each with only one nucleus. But there are some cases, you can see this in fungus, some muscle cells, where you will actually have one cell that has multiple nuclei because it did mitosis, but it didn't do cytokinesis. So we will discuss them as being two separate, usually linked ideas. So during mitosis, there's going to be four phases that I want you guys to know. You guys may have seen this before. There's a phrase IPMAT that you might see someone say, interphase is the I, but then the other phases are written here: prophase, metaphase, anaphase, telophase. Now I want you guys to know the names of these phases and the basics of what happens. I'm not going to make you know every last detail. That's not something you have to know. But I do want you to have a general grasp of what the phases are, the order in which they occur, and like the major event that kind of goes on there. So the first phase that we're going to see is going to be prophase. Now in prophase, the major task is we're trying to break down the nucleus and form chromosomes. We're also going to form some spindles and such that are going to connect to everything, but this is really the prep phase. So when I think about prophase, I kind of think this is prepping. If you see chromosomes, you know you've at least gotten to prophase. You could be further along, but you're at least in prophase. So the starting phase, the prep phase, prophase. Now, once the nucleus has disintegrated, once you have the spindle fibers, once you have the chromosomes, now all these things can interact. Because you can't get the spindle, these protein fibers, they can't attach to chromosomes if they're protected by the nucleus. So once we've gotten through prophase, you can now attach the spindle fibers to the chromosomes, because the spindle fibers are what's going to haul it around. It's what's going to drag the chromosomes. And so we can now get to metaphase, which is where we line everything up in the middle of the cell. You can see right here, they're lined up single file. And so this allows for us to make sure that everything's organized. Because the next step after metaphase, anaphase, is where we actually split these things. So we currently have where each chromosome is actually two sister chromatids, two copies of themselves stuck together. We only want one copy in each cell. So we're going to pull apart those sister chromatids at that little middle region called the centromere. It's a little protein. So we're going to pull that apart and pull each chromatid to a different side, or sometimes called a pole, of the cell. So that's going to be anaphase. But it's critical in metaphase everything's lined up properly, because if we screw this up, if we screw up anaphase that follows and we accidentally grab too many or too few of the chromatids, you typically get disorders or death. So it's kind of important that we get this metaphase and anaphase step down perfectly. So after anaphase, after we've kind of dragged stuff to either side of the cell now, so each side gets its one complete copy of all the DNA, we can then move into telophase. And telophase, I just remember where it's the opposite of prophase. So in prophase, we were kind of forming spindles, forming chromosomes, and getting rid of the nucleus. Well, in telophase, we're going to be getting our chromosomes to go back to being that loose chromatin, so where the DNA is not tightly wrapped. We're going to go back to having no spindle fibers. We break them back down, and we're going to go back to having a nucleus. We reform that nucleus around our DNA protectively. So telophase also will oftentimes have the start of cytokinesis occur. So like in this case, you can see telophase is essentially here where the nucleus is reforming. But a lot of times too, you'll see people draw telophase with some of this cytokinesis starting to happen. So don't freak out. Because we think of mitosis and cytokinesis as being two separate processes, they can actually overlap. So starting in anaphase, you might even start to see some of the effects of cytokinesis. Just don't worry about it. Just realize, yep, cytokinesis is going to happen. It's going to start happening somewhere around anaphase, telophase typically, and it'll finish after telophase is done. So without further ado, let's wrap up cytokinesis. So this is the last part of the M phase. Sometimes you'll see it written as its own phase as a C. And so this is going to be how we split the cell or the cytoplasm into two. Now there's two ways to go about this. The first one is if you don't have a cell wall, so you don't have anything rigid surrounding you, you can do what's called a cleavage furrow. And now a cleavage furrow is going to be where we take some of these protein filaments called microfilaments. So these are just like long ropes of protein. And we can make like a lasso. And so that lasso will go around the middle of the cell, and then we just tighten it. And so by tightening this lasso, these filaments, we can start to drag the cell in. We can shrink it until eventually we pinch the cell in two. So eventually we get something that looks like this as it pinches apart. So if you see cleavage furrow, I mean, come on guys, this should be one of the easiest things in the world to remember. Cleavage furrow, let's be honest, it looks a little bit like cleavage, all right? So that just means to pinch apart, but it only works if you don't have a cell wall. If you have a cell wall, we can't pinch apart. That cell wall is too rigid, it won't allow it to. So in that case, we have to do something that's building a cell plate. So this is what's going to occur in anything with a cell wall. Stereotypically, we'll say plant cells, but it could be algae cells. It could be even some prokaryotes will use a similar thing if they have a cell wall. And so this is where we're going to build new cell membrane and a new cell wall in the middle. So instead of pinching, if you think of it like a room, if you're trying to divide that room, you build a wall down the middle. We haven't ultimately pinched the room or flexed the room at all. We just added a new divider, which gives us two cells at the end when we finish building the cell plate. And so you'll have the cell wall in the middle with cell membrane on either side. So that way it's now two complete separate cells. So cell walls means you use cell plates. If you don't have a cell wall, you use a cleavage furrow.